Hello, I'm Luke Ternes and I'm from Oregon Health and Science University. Today I'll be walking you through my poster covering a variational autoencoder architecture that we've designed and implemented in order to control for undesired features in single-cell imaging data. Single-cell quantification and analysis are becoming increasingly necessary because they allow us to glimpse the diverse heterogeneous nature of cell state. At the same time, multiplexed imaging devices that are emerging, such as cyclic immunofluorescence and multiplexed immunohistochemistry, allow us to get a better glimpse and a better representation of these cell phenotypes. Now, once we have these images, it's important to extract meaningful features at the single cell level, and variational autoencoders have been developed to do just that. The problem is that variational autoencoders can overanalyze and can be lazy and only pick up features, uninformative features such as rotation or size, and methods have been developed to counteract that model laziness, and modifications and novel architectures have been developed to do just that. However, most of these methods are targeted toward a single feature and usually require modification to analyze additional ones. This can be problematic in data sets like single cell, where there are multiple different uninformative features all in the same image, hindering our ability to analyze them. Here I show an example of what I was just discussing before, where a variational autoencoder performs lazily, only extracting key uninformative features and ignoring features that might be more interesting and more important for reconstruction. In this example on the left here, looking at the principal walk of reconstructions, you can see that the variational autoencoder learned to reconstruct the rotation angle of the cell, which did vary across the dataset, but ignored some more interesting features. And on the right, you can see the same thing, where it learned to reconstruct polar orientation, which varied across the dataset, but didn't learn anything more interesting such as texture or patterning or distribution. So in order to get the VAE to extract something more biologically interesting, we needed to redesign the VAE architecture so that it put more emphasis on these other features and forced it to ignore these uninformative features. Here we show 3 of the different models we tested in order to create the results that you will see going forward. The first one is just the vanilla VAE, where an input single cell image is encoded into a latent vector, which is then decoded into its reconstruction. The second is a VAE with a corrected output, where the input image is a randomly transformed image based on a known transform for an unimportant feature, such as rotation. And the third one is a multi-encoder VAE, where we have several parallel encoding blocks, each one correcting for a different unimportant feature, and a single decoder and single shared latent space. Here we show the learning objectives for each of the different architectures we used. As you can see with the vanilla VAE, the learning objective is simply the difference between the input image and the reconstruction. With the VAE with corrected output, the objective is the difference between the corrected output and the reconstruction based on a randomly transformed input, which teaches the model to overcome this unimportant feature by correcting it to match the corrected output. And third, with the multi-encoder VAE, the objective is the difference between the corrected output and several different randomly transformed inputs combined into a single latent space via a multiplication layer, creating a single reconstruction. This hereby forces the model to focus on the mutually shared information rather than the exclusive uninformative features in each image in order to create a good corrected output reconstruction. As we can see here, using a multi-encoder VAE is able to remove all of these unimportant features from the encoding space. In the vanilla VAE at the top, we can see that there is a strong correlation between the encoding space and the features of interest, both rotation and polar orientation. By implementing a rotation controlled VAE controlling for just angle, we can remove the correlation to angle but keep the rotation to polar orientation. When we use the multi-encoder, we're actually able to remove the correlation to both unimportant features, thereby removing them from the encoding space. Here we show the improvements that a multi-encoder VAE can have on a biologically relevant dataset. This dataset was obtained from the LINX consortium where cells were treated with TGF-beta and PBS and imaged after 48 hours. The vanilla VAE, as you can see on the left here, is unable to separate these two perturbations either in k-means or in UMAP space. And when we look at the regional cell images from within this UMAP space, we can see that they're only really being separated by these unimportant features. As you can see on the left, all the cells have the same rotation of facing the upper left, and all the cells in the right image have the rotation of facing the upper right, which is not biologically interesting or relevant. When we look at the multi-encoder VAE, however, we can see that both in k-means cluster space as well as in UMAP space, we're actually able to get really good separation of these two perturbation conditions. And then when we look at the regional cell images, we can see that both perturbations are being reconstructed and separated based on phenotypically relevant factors, and we're actually capturing texture and distribution from these images. All of this goes to show how multi-encoder VAEs can be applied to capture more novel and interesting morphologies and single-cell information. In conclusion, we can see that vanilla VAEs are prone to extracting biologically uninformative features, which can guide downstream analysis in ways that obscure more important information. But by combining multiple encoding blocks into a shared latent space, we can encourage the models to focus on more important and biologically interesting features, which can improve our downstream analysis and give us access to more novel morphological and biological features. One limitation of this, though, is that all the unimportant features that need to go into this need to have a known transform so that they can be randomly transformed into the input spot. This work was supported by the Cancer Systems Biology Consortium, the Human Tumor Atlas Network, and with data supplied by the LINCS Consortium. Thank you.